Hey everybody, it is your life science / biology teacher, Mr. Poser. Today we are starting a new unit. It's called Unit 6 on gene expression. And our first topic that we're going to get into is DNA structure and replication. So our last unit, Unit 5, is on the cell cycle, and a big part of that was mitosis. And mitosis is the process by which a cell has to first, copy its DNA and then split evenly so that each of the new cells has an exact copy of DNA. And that's going to be really important because every single cell needs its DNA, and today we're going to see why every single cell needs its DNA, what's its purpose, and in order to understand its purpose and its function, we have to understand its structure, and we're also going to get it a little bit into how it's able to copy itself, which is a super important part of the cell cycle. All right, so the first question I have for everybody here, just to keep in your back of your mind, an analogy here. If you've ever bought furniture online or you've bought it at IKEA or something like that, you may have had to assemble it once it reaches your house, right? And you have to put it together so that it's correct and it works properly, right? So let's just say you bought a chair. Could you assemble that chair without instructions? Maybe some of us could, but how would that feel? How would that look? And why if you didn't have the instructions to build that chair? And the second question I have, the second analogy that I'm going to make is like, okay, how is heredity, which we're going to talk about here in a little bit, like a family recipe? Right? My family recipe, the first one that comes to my mind is Poser mushrooms, right? So you saute the mushrooms and then you add a boatload of garlic and a bunch of Tabasco sauce and stuff. And that's kind of like a family recipe. How is that like heredity, which what we're going to talk about here in just a second? And it does have to do with DNA. And so we're going to come back to that. But let's talk about you for a second. You are 100% unique, meaning that there has never been a living thing, nor will there ever be someone who is exactly like you. Your set of traits was 100% unique. Nobody or nothing will ever be exactly like you. And never has there been anybody exactly like you unless you're an identical twin. Hey, so if that's uplifting, great, because you are special and you are unique. Yes, you, talking to you. And I mean that literally, because your traits, you have a different set of traits or distinguishing characteristic than anyone else who has ever lived or will ever live. You are completely unique. Nobody else is like you. So where exactly do you get those completely unique traits from? How are you unique, and where do you get that from? Well, you might already know that you get your traits from your biological parents and your early ancestors, right? So let's say you have a genetic deck of 52 cards, like there's 52 cards in a playing deck, right? 26 of those cards were a random combination from your mom, and 26 of those cards were a random combination from your biological dad, right? So, and your deck of cards is a combination of both your mom and dad's. It's a random combination of both your mom and dad's. And your siblings, if you have the same mom and dad, have a different combination of your parents' cards as well, a different combination of their traits and their genes, right? Your heritable traits are a unique combination of your biological mother's and your biological father's traits. But how did they give you those traits? Did they say, here's my 26 cards, here's my blue eyes, here's my brown hair, and I don't know, big nose or something like that? I don't know, I kind of have a big nose. But how did they actually give you those traits? How is that even possible? We've known for thousands of years that, if you have kids, your kids end up looking like you and acting like you and being like you, right? But for a long time, not until the 20th century, did anybody know how this actually occurred. How does heredity actually occur? And it's through this molecule. It's called DNA. Okay, in our previous unit, in Unit 1, we talked about carbohydrates, proteins, lipids, and nucleic acids, right? And DNA is the most important nucleic acid. This is the molecule, not those other three, that gets passed down from one generation to the next. And what it does, its job is to store the genetic information for all organisms. That's right, every single living thing. It doesn't matter if you're a mushroom or an amoeba or an elephant or a cactus, okay? You have DNA. And every living thing shares a little bit of DNA. So we're all one big family. We'll talk about that in another unit. But anyway, all living things have DNA, and DNA is heritable. It is the genetic material that is passed down from parent to offspring. It's what determines you, right? So your set of instructions that you got that help your cells to build you, which is what DNA does, is a little bit of your, it's about 50% of your mom's instruction book and 50% of your dad's instruction book. It's just a different combination of chapters from that book, okay? So like I said, it's your instruction book on how to build you, and this is kind of what DNA looks like, and we're going to be talking about that on a molecular level here very shortly. But first, let's talk about one of the main ideas of all of biology. It's called the central dogma of biology. What DNA's job is, it contains an information code cells use to make RNA, and that RNA is used to make proteins. And again, calling back to Unit 1 here, different types of biomolecules. Proteins are the types of biomolecule that do just about everything. But it's not the proteins that are passed down from one generation to the next. It's the instructions to make proteins that are passed down from one generation to the next. And this topic of this unit, the unit title is called Gene Expression, and this is a very simple schematic of what it looks like. DNA, which stores this code, this instruction book to build you, is transcribed into a smaller molecule called RNA, and then that transcript is translated into a code that's used to build proteins, or that code is used to build proteins. All right, and proteins do just about everything. This idea right here is called the central dogma of biology. Central meaning like super important. Dogma means a set of principles that all living things abide by. Information in DNA is used to build RNA, and then the information in RNA is used to build proteins. Okay, so what's going to happen here is that a cell will take a gene, which is just a short segment of DNA, open up the DNA, transcribe it into this messenger molecule called RNA. Okay. That process is called transcription, and then RNA is sent out of the nucleus, okay, and then carries a message and it carries a code in order to build proteins, okay? Because really, if you boiled it down, what does DNA do? Yeah, it makes up you. It's your genetic, it's your molecular fingerprint, all that stuff. But all it is instructions to make proteins. Once we boil it down to the molecular side of it. And this is the central dogma of biology. That means every living thing has DNA, makes RNA with it. It's through transcription, okay? And then transcription is used, or that RNA transcript is used and sent out of the nucleus, or not in bacteria, but it's the RNA transcript is used to make proteins. All right, every living thing has to do that. Okay, and I just want to shout out, we don't have to do the whole history lesson right now because we're a little short on time, and we want to get to the real gist of this here. But it took many years and very many people earning Nobel Prizes to figure out that this central dogma of biology here is that DNA is the genetic material. It's used to make RNA, and RNA is used to make proteins. It took a lot of very many famous scientists and very many famous experiments in order to figure that out. So I just want to shout them out here, and we're going to move past that. Shout out Rosalind Franklin especially. All right, so then what is DNA? How does DNA actually work? DNA's structure allows it to store information and to be read. DNA is a polymer, which means it's a huge molecule, but it's made up of tiny littler molecules called nucleotides. And nucleotides are what we call monomers. If you remember, once again, to Unit 1, all of those macromolecules or polymers have littler molecules that make them up called monomers, right? And the monomer of a nucleic acid here in DNA is called a nucleotide. And there's 4 different types of nucleotides that we're going to be looking at here in a second. Okay? But DNA is 2 very long chains of these molecules called nucleotides. And there's only 4 different types of nucleotides. And those are A, C, G, and T. Okay? So DNA is it's a double polynucleotide strand. Okay, we're going to look at that in just a second here, but the molecules that make up DNA look like this. They're called nucleotides. Okay, and depending on what this, what's called the nitrogen space looks like, it determines whether or not it's adenine, cytosine, guanine, or thymine. Okay, A, C, G, or T. Okay, so these are going to become very important here in a second because these are like the letters in the language of life. Okay, if you have an instruction book, it has to be in a particular language, right? And the language of life is only made of those 4 letters. Imagine having your instruction book trying to assemble that furniture, but the instruction book only has 4 letters in it, right? English has 26, and we have like a whole bunch of other symbols that we use to express language in writing, right? Or in speech, and DNA only has 4. Okay, so you have to have a lot of those letters in order to get that message across. And that's why DNA is billions of these nucleotides long. Billions, literally. Remember in our last unit, if you took DNA from one end to the other in a human cell, it'd be 6 feet long, right? So there's so many of these nucleotides and so many letters in order to build that code. Okay, and how these letters are arranged, they're in a double helix or a twisted ladder. Okay, they kind of look like my molecule right here. This is DNA, right? You've probably seen it before, but you just didn't realize that, the significance of the double helix shape. Okay, so there's 2 strands here of nucleotides, and they kind of meet up in the middle, kind of like a ladder, right? So this picture over here looks like a straightened out ladder that you could climb up to get to the roof of your house or something like that, okay? But then DNA is kind of twisted, like a twisted ladder, and that's what we call a double helix. There's two different strands. It says each molecule has two strands of nucleotides that form bonds in the middle, more particularly hydrogen bonds. Okay, so these two strands meet up in the middle, but they meet up in the middle in very, 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 very predictable patterns. And those are what we call base pairing rules. Okay, nucleotides always pair in the same patterns. These are called Chargaff's rules. Okay, so an A nucleotide, remember we only have 4 letters, A, T, C, and G, but they always match up with each other in predictable patterns. An A nucleotide will always match up with a T nucleotide. And a, C nucleotide always matches up with a G nucleotide. Okay, so the apple always goes in the tree, and the car always goes in the garage. That's how you can remember Chargaff's rules, so your base pairing rules, right? So even at this molecule over here, A matches up with T, C matches up with G between both of these strands of nucleotides. Okay, so if this is one strand of DNA, take a second and see if you can't do it yourself. Can you pair the nucleotides of this DNA strand? Okay, if you, I'm not showing you the other strand, but I bet you can figure it out if you know your base pairing rules. Car goes in the garage, apple goes in the tree, right? So pause if you want to try it yourself, but if not, I'm going to press on here. All right, so here's what the other strand would look like. Okay, check it out. Car goes in the garage, apple goes in the tree, car goes in the garage, apple goes in the tree, okay, all the way down here, okay, so that each one matches up and forms the complementary pair with the other strand. Now, this is going to become very important when we talk about replication. All right, here's another DNA molecule, and once again, it's two strands of nucleotides that bond in the middle and form predictable patterns, A and T and C and G, right? So here's what DNA looks like if we really, really get down to the atomic level even, okay? And they bond in the middle. So DNA replication, how it's copying itself, it takes advantage of these predictable rules that A always goes with T and C always goes with G. DNA replication, or the copying of DNA during the cell cycle, occurs by separating the two strands and making new ones by matching up nucleotides, right? So in order to make a copy of the DNA, it doesn't just come by and copy the whole thing. It splits them apart first and then makes a copy, just like we just did, off of the old existing strand. And you'll see I have a nice schematic here that we'll use in just a second. Okay, and so in order to make a copy of DNA, we need to split up those two strands, and the enzyme that's going to be doing that is called helicase, the enzyme that unzips the DNA strands to be replicated. All right, so here's our double helix. Helicase is going to separate them out, and then a new strand is going to be built off of the old one according to our base pairing rules. A goes with C, or excuse me, A goes with T, and C goes with G. Right? So here's what this is going to look like, and I'll show you in just a second. But then these special molecules called DNA polymerases then add new matching nucleotides to the template strands to make new complementary strands of DNA. So first DNA has to be unzipped, it has to be separated out, and then we take advantage of, DNA polymerase takes advantage of the base pairing rules, A goes with T, C goes with G, to make new strands, and then you have an exact copy of your DNA. Okay, so this is what it looks like. Here's our two old strands. I have those in black, and then the new strands will be in blue. All right, so here's what DNA replication looks like. Here's the same exact set of nucleotides that we had earlier. There's the complementary strand right along with it. Helicase is going to separate out those two strands so they're not bonded to one another anymore. Okay, and then DNA polymerases, those enzymes, are going to move kind of like trains along a train track along the old strand to add new nucleotides to it, and they're going to follow the rules here. So a G is going to go there, C is going to go there, C is going to go there, T is going to go there, A is going to go there. They're going to follow the rules and follow the template strand to make those brand new strands. And that's how DNA copies itself. Okay? That'll be it for this video. Please let me know if you have any questions, and we will see you